Good morning. Thanks for joining us for the webinar this morning. We're going to hold out for a few minutes here to let some more people join us before we start on our first presentation. Okay, so good morning. Thank you for joining us for the second of our webinars that we're calling Sharpen Your Dairy Skills as we Flatten the Curve. Hope everyone out there is doing all right in this crazy time. This morning, I'm really pleased to have our first presentation from Dr. Angel Abuelo. Dr. Abuelo grew up in the northwest part of Spain. Did his veterinary degree as well as his PhD at the University of Santiago de Compostela there in Spain. Spent some time in the University of London and joined the faculty in the College of Veterinary Medicine at Michigan State University just over 2 years ago. And from my standpoint, has been doing an excellent job both on the research and the extension side for us on the dairy team at Michigan State. So this morning, Angel is going to share with us some sort of newer research on immunity in calves and some of the work he's doing. So Angel, thanks for being a part of this.
Thank you, Barry. I'm very pleased to be with you all this morning. And yeah, so as Dr. Bradford was mentioning, like my goal for like this kind of like 25 minutes is to just go over some of the new research on ways to modulate or improve the immune response in calves. Obviously, this is not going to include all the potential ways or alternatives to modulate immune response in calves. So there are like different ways that unfortunately we're going to have to lift out, but hopefully it will give you an overview of some of like the new research and some of the work that I and others have been doing in this area. So the outline for like this short presentation is really just to kind of like summarize again, like why it's important, why should we care about neonatal calf diseases, and then put everyone back together into what like the basics of immunology in calves and how their immune system works and how it's somewhat different from what we see in adult animals, and then go into the different ways of research that we are doing to see how we can improve those immune responses. So as I was mentioning, like neonatal calf diseases are really important. Like the data from the last NAMS report still puts morbidity kind of like 33%. So it's kind of like 1/3 of the heifers born in the US would experience some disease during the pre-wean period, and mortality is about like 6%. And this is mainly due to infectious diseases such as diarrhea or pneumonia. And yeah, as you can see, like those, a major contributing factor for those infectious diseases is like the inability of the immune system of the calves to mount an effective immune response against those pathogens. Also really important to remember is that rearing costs usually represent 15 to 20% of total production costs. I know that those estimates are somewhat old. As you can see, like that research was done in the early 90s. So it will be interesting to see what it represents right now. And obviously it's going to depend on every farm. So, but still it's a significant percentage of the costs associated with milk production. And evidence is that reflects the fact that if a calf gets sick during this neonatal phase, that has a lifelong impact on their productivity and health status. So if a calf gets pneumonia or diarrhea when they become a heifer or a productive member of the society of the milking herd, then their productivity is going to be affected and their risk for subsequent disease is going to be increased. So it's also something to bear in mind. Then when we talk about immunology in calves, we always have to look at it from like the two perspectives of passive immunity, which is that is transferred from the dam to the calf, primarily through colostrum, because the placenta of ruminants does not allow for transfer of immunoglobulins in utero, and then the active immunity, which is the protection that's conferred by the animal's own immune system. For the today, we are going to focus primarily on active immunity, on how we can improve the responses of the immune system of the calves, but bearing in mind that management and colostrum is paramount, it's really important. So there's not really like a magic bullet that's going to correct or improve poor management. So yeah, colostrum is really, really important. We are just not focusing it right now. I'm just leaving there the link and the QR code for some of the colostrum management guidelines that the dairy team at MSU Extension has put together, some like more like the updated information. So if you're interested in that, please read and reach out if you have any questions. But yeah, so we are going to focus primarily for this talk on the active immunity side. And we always, when we talk about immunology or the immune response, we kind of have tried to put the immune responses into different boxes, and they're like different classifications. One of them is kind of like we talk about innate immunity, which is like the first responders, or it's a generic response to when the immune system faces a pathogen, but it has no long-acting response. It's a really quick response, but it kind of like reacts the same way, whether it's an E. coli or a salmonella. So it's really non-specific. And on the other side, we have like the acquired immunity, which is specific to the pathogens. And it does have a memory, but it requires time to develop. And then both innate and acquired immunity are going to have both cell-mediated factors and, well, what is called like humoral factors, or more like soluble factors. So again, innate and acquired, both cell-mediated and humoral in each of them. And again, we try to see everything in different boxes, but they really interact among each other. So it's really important to know. So like the phagocytes, which are like the neutrophils, macrophages, all those cell-mediated components of innate immune systems do interact with the lymphocytes. So we kind of like sit in different boxes, but when they kind of like act synergistically when facing a pathogen. but for the purpose of kind of having a clear picture, we still have it that way. So now talking about the newborn calf and their immune system. So in theory, unless the calf has been infected in utero, when a calf, when it's born, has no or non-detectable concentrations of antibodies or immunoglobulins in serum. That's why the transfer of passive immunity through colostrum is really important. So, okay, so a calf is being born with no antibodies. Then other issues that we see on neonatal immune systems is that the activity of the complement, which is the humoral component of the innate immune response, it's also lower than adults, and the cell functions of those immune cells is also decreased compared to adults. And it has been shown for like neutrophils or macrophages, like those phagocytes or first responders, but also for other antigen present in cells and B and T lymphocytes. What is important to remember is that calves do have an immune system. So they have like the immune cell machinery of a mature immune system at birth, but that immune system is just naive and usually in a quiescence state. And the development of the immune system really begins when the calf is in utero. So when the development of the thymus, or where the development of the T cells takes place, it's already evident at 40 days of gestation. So when we are already doing like preg testing and see like something that's about like 2 centimeters in length, they are already starting to develop their own immune system. So it is not true that calves are born without an immune system. It's just that their immune system has not been primed and it's not strong enough, if we want to say it these days. And we also have learned from different infections, from different viruses, particularly bovine diarrhea virus, that the fetuses can respond to some virus as early as 70 days of gestation. And by the 3rd trimester, the calf can respond to many different infections. And if that infection takes place, that's when the calves would have antibodies at birth before they ingest colostrum. So they have antibodies that they generated themselves. And another thing to consider is that within like the development of the immune system in the fetus, the biggest expansion of immune cells takes place during the last month of gestation. Something that I became interested on, it was like, well, if all those that immune development takes place in utero, where they are really exposed to some extent to the same factors of their dams, how that has all that kind of like maternal stress or effects in the utero affect their immune responses. So that's one of like the studies that one of my veterinary students did for her summer project, in which we evaluated the effect of metabolic stress, which we usually define as a combination of excessive lipid mobilization, inflammatory and immune dysregulation, and oxidative stress, which is like the imbalance between pro and antioxidants, or like free radicals and oxidative damage to cells. How that we know that happens in cows when they transition, so when they, even like during the dry period. So how would that we know that affects the immune responses of cows, whether that can have an effect on the development of the calf during late gestation. And this was like a really preliminary study, but what we found was that calves that were born to cows that experienced more, we measure oxidative stress through an indirect measure, so which is kind of like this oxidant status index. But so calves that were born to cows with a higher values of oxidant status, so that they have a higher proportion of free radicals to antioxidant defenses, had lower birth weights and lower body weights throughout the first 4 months of life. And the same for cows that mobilized more NIFAs during the last month of gestation. So calves that were born to cows that had more lipid mobilization had lower birth weights, and those lower birth rates were maintained at least throughout the first month of life. We did not find any difference on average daily gain. So that means that it's not really a compensatory growth, at least during the first 4 months of life, 4 weeks, of life in those calves. So yes, it does have an effect, at least on the development of the calves. And we also tried to assess their the immune responses of the calves. So since this was a preliminary study, the only thing that we just did was kind of like an ex vivo stimulation assay. So we collected whole blood from the calves, kind of like faked an infection in the blood and see how that how those cells responded. And we measured it through like the release of TNF alpha, which is kind of like a pro-inflammatory cytokine. And what we saw was that calves that were born with cows that experienced more oxidative stress had less robust responses to that stimulus, as you can see by the black bars. And the same results were found for calves that were born to cows that experienced higher degree of inflammation during the last month of gestation. So it seems that, yes, the status of the dam during that period, during the dry period, is going to affect the immune responses of the calves. Whether that effect has enough entity to actually affect disease susceptibility is something that we still are trying to find out. So we know that their immune cells are not responding as robust, but whether that's protective, that difference is protective enough for disease, it is something that we are still determining right now. And in fact, that's something of what my lab is currently working on, is to study really the clinical impact of that late gestation metabolic stress. So we were able to find something measuring in the lab. Now does it translate to improve health in the calves? And another of the studies that we are currently still running, despite all the COVID-19 issues, thankfully, is trying to study the effect of different strategies. In this case, we are doing a nutritional intervention to decrease oxidative stress, and see how that's going to affect the immune responses of the calves. Another important, well, as I mentioned before, the innate immune system of the calves is also immature at birth. And looking at neutrophils, which are some of like the cells that first would respond to an infection. So we know that the oxidative burst, which is one of the ways neutrophils kill bacteria, is reduced during the first week of age, and the activity of metalloperoxidase, which is also another way through what neutrophils kill bacteria, it's also reduced. And those levels do not really reach like the levels of adults until puberty. So it takes significantly long time for the innate immune system to actually work as effectively as in an adult cow. So looking at neutrophils, one of the factors that has been studied in neutrophils is like the use of colony stimulating factors, which are naturally produced cytokines that stimulate the production of neutrophils and also improve their function. And we were interested in that because there is a drug that is approved for improving neutrophil function in transition dairy cows, which is it's a colony stimulating factor labeled Imrestor from Elanco. And we were wondering whether, okay, has been approved for cows that have neutrophil dysfunction around calving, but calves also have decreased immune function, neutrophil function. So we were wondering whether that drug could also potentially have a positive effect on calves. And at the same time, probably that we started with our study that I'm going to talk in the next slide. There's another group from Brazil that actually administer the drug, which is like this pegbobigastrem, to 10-day-old calves. And they only measure population of cells, not really their function, but they saw that after the administration of this drug to the calves, they saw an increase in the number of phagocytes in those calves. So yes, it does increase the number of neutrophils. However, what we were really interested was about like the function, not necessarily the numbers, because as I mentioned before, calves are born with the immune cell machinery, is their function, which is not optimal. So we really were more interested in see whether it can improve the function rather than just numbers. And when we think about like the different functions or ways by which neutrophils can kill bacteria, that's what have here in like this kind of like graphical representation. So when neutrophils are exposed to bacteria, one of the things that they do is first they engulf the bacteria, which is what it's called like the phagocytosis, and then they would kill those bacteria either through like what's called like the oxidative or respiratory burst, in which is like they just kill the bacteria through an overload of free radicals or reactive oxygen species. They will also use like degranulation, so they accumulate myeloperoxidase, which is also a molecule that will produce free radicals to kill that bacteria. And then lastly, it's what it's called like the netosis, or the formation of neutrophil extracellular traps, which is when the neutrophils are about to die, they kind of like not really explode, but they release their DNA and different proteins and form kind of like these threads that then they will trap the bacteria in there. So those kind of like the main microbacterial functions of neutrophils. So we wanted to explore whether using colony stimulating factors, we could improve those microbacterial functions in neonatal calves. And rather than going first to the animal, we decided to run an in vitro study in which we isolated the neutrophils from 2 to 3 day old calves, and we kind of then treated them with colony stimulating factors in vivo, and we measure the activity or those microbacterial functions that I was mentioning before. And interestingly, what we found was that, well, you can see here on the blue bars are the neonatal calves. We use for comparison also neutrophils from mid-lactating cows, so that we have like kind of like a reference for what a mature immune system will, how a mature immune system will respond. So that's what we compare. So as you can see here, like calves are always have like a lower values of those functions than cows. But what it was really interesting, for example, looking in here at the phagocytosis, is that, okay, at concentration of colony stimulating factor 0, which is that was our control, the phagocytic capacity of neutrophils from newborn cows were lower. However, when then, and regardless of the concentration of the colony factor use, it significantly increased, and it actually increased to levels that were comparable to those of a mature cow that had not been exposed to the colony stimulating factors. So for the release of myeloperoxidase or the degranulation and phagocytosis, yes, colony stimulating factors had improved those functions, and in both cases to levels comparable to a mature cow. However, we were not able to identify difference on the respiratory burst capacity or the capacity to form extracellular traps. So we did see some effects on, again, this was kind of like a preliminary in vitro study in some of those key microbacterial functions, but not in all of them. But this still suggests that we probably can explore further and now see whether that in the calf would have an effect in terms of reducing disease risk. Another really interesting aspect that I'm passionate about, probably because it was like what I concentrated in my PhD on, was its oxidative stress. And oxidative stress, it's usually like defined as like, again, the balance between the total amount of oxidants, some of which are free radicals, and we usually call them like reactive oxygen species and nitrogen species. And it's like the proportion of those against antioxidants. There's kind of like a balance between free radicals and the defense capabilities of the antioxidant systems in the body or the cell. And that's usually referred to oxidant status or redox balance. And when there is an imbalance, when the production of pro-oxidants or accumulation of pro-oxidants overwhelms the capacity of antioxidants, that can lead to oxidative damage to macromolecules that would impair the function of cells or different tissues. And we know from humans and research that has been done in adult cows that oxidative stress affects the function of immune cells. So they decrease the immune cells. And there is also evidence in cows and in humans that it's also linked to an increase in the risk for disease. However, there's not really any data available in calves, but one of like the early studies that were done about trying to characterize oxidative stress in calves that I kind of like, it was like a summer project for a student that, and it was like really interesting because it was around the time that I was finishing up my PhD. And I think that I had these samples analyzed 4 times because at the beginning I wasn't believing them. So this like the oxidant status index, again, to interpret it, the higher the value, the more pro-oxidants to antioxidants, or the higher indirect oxidative stress. And what we found, like those values, so what is represented by this gray dash line were the max values that I was seeing in transition cattle during my PhD. And as you can see here, throughout the first month of life, calves had higher values, which at the beginning didn't really make much sense to me. It was like, how is it possible that calves have more or higher values of oxidative stress than transition cows, that we know that they do experience oxidative stress? And then digging into the literature, just started reading, okay, well, if you're feeding like milk replacer and things like that, we usually do not balance as well the amount of antioxidants and vitamins and things like that in the diet of newborn calves. But they are also exposed to high metabolic demands if they are growing as they should. So it was really interesting to see, okay, what are really the effects of this increased period of oxidative stress in newborn calves? How is that affecting their immune responses, since we know that that affects the immune responses in other animals? And this is, again, something that also my lab is working on right now. So we know that lymphocytes, which are our acquired immune system cells that actually would be useful both for T cells and B cells, so both humoral and cell-mediated immunity, when they are exposed to an oxidative challenge, as in this case, we have like here, like different changes of their apoptosis is going to increase, so they are going to die faster. Capacity to respond to stimulus is going to decrease. Their capacity to proliferate is also going to decrease. Their capacity to synthesize immunoglobulins is going to also decrease. And as you can see, many of those functions are essential if we want the vaccines that we are using in those calves to work. So this is something one of my PhD students is working on right now. And again, we are starting first in vitro. And what we did here is like we isolated PBMCs, or immune cells, from blood from calves that were 4 to 5 days old. And we have like an in vitro oxidative stress model. So what we do is like we add to the media of those cells this AAPH, which is a generator of free radicals, and we have like the concentration titrated so that we know that the cells under those conditions are oxidatively damaged, but their viability is still high. So they're still alive, but oxidatively damaged. And then we kind of like expose them to bovine herpes virus 1 challenge. And here you can see that the PBMCs that were isolated, that were exposed to this virus, were able to produce a considerable amount of herpes virus specific antibodies, but that capacity decreased under conditions of oxidative stress. So yes, also in calves that are exposed to oxidative stress, their capacity to synthesize immunoglobulins specific to the pathogens that they are facing is decreased. And that's also the case for the activation of both regulatory and cytotoxic T cells after viral exposure. So in summary, yes, it seems that oxidative stress during this period is negatively impacting the capacity of calves to mount an immune response. So what we are trying to see now is like, okay, what would be like the ideal or a good combination of different antioxidants that we could use in these calves to improve their immune responses and then move on to in vivo trials in which we will assess that in the live animals. And when we think about antioxidants and how we can supplement them, probably we kind of have that somewhat figured out, although not perfectly tweaked in adult cows. But if we think about, okay, can we supplement the cow so that the calves would be born with a better antioxidant capacity? So maternal supplementation of with antioxidants or during gestation, it's very common in humans. And we know that if we supplement the cows during the dry period, that's going to transfer to the calf. But we have to remember that the placenta of ruminants will limit which antioxidants are transferred. For example, selenium is transferred via the placenta, but other vitamins, for example, are not. And it's also important to remember that we cannot just go and feed whatever we want, because excessive supplementation during late pregnancy has been linked in some studies with fresh cow disease. So it's kind of like a fine balance between how much it's beneficial and when it starts becoming harmful. And it has also been linked to the stillbirths in humans. So probably the potential for that to also happen in cows, and probably Dr. Bradford could hypothesize how that could be linked through decreasing different inflammatory pathways that are needed for parturition. Another common practice that is done a lot of times is the supplementation of calves. And at many farms now supplement calves at birth, either with vitamin E and selenium or different minerals. However, it's like some research that shows that probably calves need far more support than that. That's good, but sometimes it's just what it's needed to counteract the oxidative challenge that birth is. And some studies can show that that's probably sufficient or improves the antioxidant status for just about a week. So probably we need to supplement more those calves. And like some other research that has been done in the last years show that, for example, the parental supplementation with trace minerals at 3 and 30 days of age improve the neutrophil function in calves, and also some antioxidant capacity, and decrease the morbidity in those calves. And when calves were supplemented again with the same trace mineral complex with a viral vaccine at a month of age, they showed an improved cell-mediated immune response and some improvements on the pathology level on that challenge. A really interesting study that was published late last year, and this was a study from New Zealand that enrolled almost 1,000 calves in different farms, showed that supplementing calves at birth decreased both morbidity and mortality significantly. However, as you can see, the values of morbidity, on the control group, are about 15% for morbidity and 12% for mortality. So they're relatively high, and in those cases, usually an intervention will be easier to find an effect on the decrease. So it might not work on every single farm. And as I mentioned before, again, management is key in those situations. So summarizing the antioxidant supplementation for calves, right now, we don't have guidelines for what should be the adequate value for optimized performance in calves. But we have to be careful right now with supplementing above levels that have been deemed safe by the literature or the scientific evidence. A, because there are some potential adverse effects of eating excessive antioxidants, and B, because some of those micronutrients with antioxidants capacity that we regularly use are linked to toxicoses, such as selenium. And as I mentioned before, selenium is transferred via the placenta, so we have to somewhat be careful, because if we are already supplementing the calves with selenium and that gets transferred through the placenta to the calf, and then we over supplement with selenium the calves, then the risk for selenium toxicosis is high, and it would be high on those animals. So maybe we need to use other micronutrients that also have antioxidant capacity. And the last thing that I wanted to talk about with regards to vaccine modulation of the immune response is vaccination. because it has been somewhat controversial, the use of parenteral vaccines in the face of maternal antibodies, and that's because it was traditionally associated with a failure to seroconvert or to show an increase in the titers of antibodies after vaccination. However, that's now we know that's not a good indicator that yes, there is not an increase in the antibody response following vaccination in face of maternal antibodies, but there is an increase on the T cell response. So, okay, we don't have that humoral response, but we still have an increase in the T cell response. And that's when we think about like signs of disease, and this is a challenge model, we can see that it's actually like protective. So vaccination in face of maternal antibodies is it's possible. It's just calves would not seroconvert. However, it's true that the magnitude of the response is going to be linked to the amount of bacillus transfer antibodies. So it's something that needs to be considered on a farm-to-farm basis. And yes, probably all of you are thinking, okay, we are just using intranasal vaccines to overcome that maternal antibody interference. But and it's really good for particularly like for respiratory pathogens, because we are creating localized immunity in the port of entry for those pathogens. And yes, it might be superior to injectable vaccination. However, some of the earlier studies shown that duration of immunity might be lower than parenteral vaccines. And in some studies, they show that that's only like probably a few weeks. So the increased concentration of, in this case, immunoglobulin A in the mucosal surface might just be shorter in time than other ways. And this is, again, a really recent study from this year from the group at South Dakota University, in which they vaccinated in face of maternal antibodies using a modified live vaccine, and all these calves have adequate passive transfer of passive immunity, so they did not have failure of passive transfer. And the interesting part was that then they did a challenge with syncytial virus, and you can see here that the vaccinated group shed less viral particles than the unvaccinated ones. And interestingly also, that parenteral vaccination with a modified live vaccine actually increased mucosal immunity in those calves. So the concentration of immunoglobulin A in the nasal secretions was also higher on the vaccinated group. So yes, we can also elucidate a mucosal immune response by the use of parenteral vaccines, even in the face of maternal antibodies. And with that, I just want to summarize right quick that, again, the immune system of calves starts developing in utero. So dry cow management, it's important, and we need to take into account that we are not only preparing the cows for freshening, but also the calves that they are carrying. Calves do have an immune system at birth, but it's immature and naive. It's our job to actually care for it. And it seems that immunomodulation in newborn calves is possible, but yes, probably further research that includes head outcomes, it's needed, because it's easier to find like an effect on an assay in the lab, but whether that translates to decrease morbidity or decrease mortality, it's a different story, and many studies have not focused on that. And again, vaccination in face of maternal antibodies, it's possible, and mucosal immunity can be induced by both intranasal and parenteral vaccination. However, as I mentioned, this study just came out a couple of months ago, but I think that still would be interesting to see a head-to-head comparison regarding the protection capacity. So we know that we can produce some mucosal response, but whether one way or the other it's better at protecting against disease, it's still to be determined. And with that, I would just like to thank you everyone for your attention. And before I have times for questions, I just want to quickly remind you, if you haven't received an email already, that we are already organizing the Michigan Dairy Health Symposium for next year, which will be on March 11th, and it's going to focus on calf health. So I hope to see you all there as well. And with that, I don't know if we have time for questions, Dr. Bradford.
Thanks, Dr. Boyle. Great job. Really interesting stuff. So I'll just remind or encourage people who are on this webinar to use the chat function in YouTube to send any questions. We're a little bit long on this presentation, so I'm just going to throw one quick question at you, Angel. You've been on a lot of farms in the Great Lakes region, and I know this answer would be specific to farm, but if you had to cast a broad general net and say probably what's the #1 thing that the typical farm could do that would be likely to improve immunity or health of calves, what would you say?
Oh God, that's a really tough question. And yeah, it's definitely going to depend a lot on, it's really farm dependent. Yeah, I mean, obviously colostrum is really important. But when you have that, a lot of times it's, I mean, vaccination, again, it's really farm dependent. That's a tough question to see that in general. Nutrition of the calves is also really important. This is something that I didn't touch on, and I have seen like several farms that probably could do a better job at feeding their calves. So we need to support also the immune system with nutrition. And something that really I didn't touch upon today, but if cows didn't have the sufficient nutritional status to mount immune responses, we can do whatever we want. We can give them other stuff to improve their immune responses. They are not going to have sufficient energy for that. We need to remember how fast we want the calves to grow. So if they have to face on top of that an infection, that's going to increase their nutritional demands. So looking at the farms that I've been to, that's probably something that I would say that needs to be tackled first before going into any of these other, you can change your vaccine protocol and try to do other things, but if your underlying nutritional management is not working well, vaccines probably are not going to work as well as they should.
Thanks very much. I knew you could handle a tough question, so well done. Well, thanks again for presenting that for us, Dr. Abuelo. And I think everybody in the audience knows they can get a hold of you if they have questions, and I think you'd be a great resource. So we'll go ahead and switch gears now and invite Dr. Turner Swartz to share his screen and prepare his presentation for us. So for a quick introduction, Dr. Swartz grew up on a dairy farm in Pennsylvania, did his undergrad at Penn State, and managed some dairy farms for a few years before deciding to go back and pursue his PhD, which he completed at Virginia Tech a couple of years ago. He actually joined my group at Kansas State for about a year before we both moved to Michigan State here at the beginning of 2020. So Dr. Swartz, if you want to share with us some information on ketosis and mastitis, please go for it.
All right. Thank you, Dr. Bradford, for that introduction. And thank you all for attending this seminar. As he said, I'm going to be talking about the effects of ketosis on immune function in transition dairy cows, and particularly how it relates to mastitis. So just a quick overview. As you all recall, the transition period represents 3 weeks prior to and after parturition. And during this time period, we see an increase in the incidence of disease, and this morbidity has been associated with a number of things, one of which is negative energy balance. So as cows approach calving, they start to get into an energy deficit due to drops in dry matter intake, as well as an increase in energy demand, particularly for the onset of lactation. And this becomes more and more exacerbated in the earlier stages of lactation. Also, during this time period, we see what we call metabolic inflammation, or kind of a chronic low-grade inflammatory response. And it's been shown pretty well in research that this is due to, or at least partly due to, negative energy balance. And so we suspect that ketosis plays a role in maintaining that inflammatory tone, that chronic low-grade inflammation that we see in transition dairy cows. So how do we get ketones? Well, Well, as cows mobilize fat reserves, they liberate NEFA, or non-esterified fatty acids. The NEFAs travel to the liver, where they can go through one of three different mechanisms. They can either be completely oxidized into energy, which is what we would like, or they can be reesterified into triglyceride to be stored in the liver, which is what causes fatty liver disease. Or finally, they can be incompletely oxidized into ketone bodies, such as beta-hydroxybutyrate, or BHB. We typically view these as increases in BHB or increases in liver triglyceride as an unsuccessful adaptation to lactation. So what is hyperketonemia? Hyperketonemia is the abnormal increase in circulating ketone bodies. It is defined by the measure of beta-hydroxybutyrate. We define subclinical ketosis as greater than or equal to 1.2 millimolar concentration of BHB. The incidence is variable across herds. There are some herds with quite low incidence of subclinical ketosis, but there is a nice study that came out of Dr. Jess McHart's lab at Cornell that showed 40 to 50% of postpartum cows will have at least one sample where they are subclinically ketotic. This disease has been, because it is so common, this disease has been associated with a number of other diseases, in particular displaced abomasums, but I'm going to talk more about infectious disease. So I'm going to refer more to mastitis. So starting back in the 1980s, all the way dating to present day, there have been numerous publications that have come out that have associated ketosis with the incidence of clinical mastitis as well as the severity of mastitis. I kind of like to describe the transition period as demonstrating the domino effect, where one disease leads to another disease, which may lead to another disease, and eventually leads to the cow exiting the herd. So stopping that first domino from falling, I think is key to improving transition dairy cow health. But like I said, and from the previous slide, these are associations. And because we have a saying in research that correlation does not imply causation. So if you look in the human literature, ketosis is actually quite a beneficial effect. It's been viewed that way. So the ketogenic diet is very popular. It's a high-fat, low-carbohydrate diet that has anti-inflammatory effects due to increased elevation of ketone bodies. And so when you look at the human disease models, such as Parkinson's or Alzheimer's disease, gout flares, arthritis, seizure activity, BHB reduces that inflammation, and it even improves longevity and has a reduction on anti-aging effects. So for us, this really kind of begged the question, is beta-hydroxybutyrate a causative disease mediator or an adaptive response to resolve disease? I mean, when you look at the human literature, it seems to be resolving inflammation. It seems to be a beneficial effect, but when you look on the cow side, it seems to be a causative disease mediator. It increases the risk for infectious disease. So we asked this question, and we were trying to find ways to answer this to see if BHB truly is increasing the risk for disease. So in order to understand that, we have to first realize that beta-hydroxybutyrate is much more than just a metabolite. It's more than an energy source. There's actually a few different mechanisms that it can function through. So beta-hydroxybutyrate can bind to or activate G protein coupled receptors. This one in particular would be GPR109A. It is a receptor that is on the cell membrane, and when it is activated, it has an anti-inflammatory effect. It blocks inflammation. Excuse me. There are also another mechanism called NLRP3 inflammasome. It's a intracellular sensor that exists in the cytosol of the cells. And what NLRP3 does is that it activates cytokines. It activates pro-inflammatory mediators. In particular, this one would be pro-IL-1 beta. So when NLRP3 is activated, it will cleave a peptide off of pro-IL-1 beta, which will then get secreted in its active form. So when IL-1 beta is active, it will then circulate throughout the body, and it is a known fever inducer. So it has pro-inflammatory effects. BHB inhibits this activation. It inhibits this pathway. So by doing so, it has anti-fever or anti-pro-inflammatory effects. The last mechanism would be epigenetics. So if you're really nerdy, like Dr. Bradford is, you will get all excited about epigenetics. So BHB inhibits what we call HDAC, or histone deacetylase inhibitors, or excuse me, histone deacetylase. BHB inhibits histone deacetylase, which in turn alters gene expression and alters protein function. So really what you should know from this is that it can inhibit or alter gene expression in a way to promote an anti-inflammatory environment. All right. So we tried to find different ways of answering this research question. Does BHB have an anti-inflammatory effect, and is it increasing the risk for disease, for infectious disease? So we did a cell culture experiment with macrophages. Macrophages are an innate immune cell. They are phagocyte. They kill bacteria. That is one of their main functions, is to kill bacteria. One of their other functions is to recruit immune cells into the mammary gland, such as neutrophils, to recruit other immune cells to defend against pathogens. The last thing that a macrophage does is it promotes tissue homeostasis and regeneration. So macrophages aren't just pro-inflammatory, they are both pro-inflammatory and anti-inflammatory. They have dual functions. We took mouse macrophages, it was an immortalized cell line, and we cultured them with BHB at 4 different concentrations, 0, 0.6, 1.2, and 1.8 millimolar. The 0.6 would be representative of a healthy cow, 1.2 of subclinical ketosis, 1.8 more of a hyperketonemia, maybe a clinical ketotic cow. And then we challenged them with Streptococcus uberis or not for an additional 6 hours. Strep uberis is a very common mastitis pathogen. The incidence of strep uberis mastitis is greater during dry period as well as the first couple of months post-lactation or post-calving when metabolic stress is most apparent. Okay. So here are our different treatment groups. We measured GPR109A, which is that receptor that I had talked about before that is activated by BHB. We have 6 different treatment groups. The 4 in the blue or the blue shades are the ones challenged with strep uberis at various concentrations of BHB. So 0.6, 1.2, 1.8, or the controls, which did not receive any BHB. Then we also have a control and an osmotic control. The control and osmotic control, neither one of them are challenged with strep uberis. The osmotic control it has a saline solution added to it. The reason why it exists is because BHB, when you add it into the cell culture media, it is added as a salt. So it has a lot of sodium in it. So in order to make sure that that sodium is not having an effect, we have an osmotic control. What I want you to get out of this slide is that strep uberis increases GPR109A. This is the receptor that can be ligated or activated by BHB to have an anti-inflammatory effect. So moving forward from that, BHB promoted an anti-inflammatory macrophage response. So we measured multiple cytokines, but the 2 I'd like to point out are interleukin 10, which is an anti-inflammatory cytokine, or tumor necrosis factor alpha, TNF alpha, which is a pro-inflammatory cytokine. When you look at IL-10, we had a dose-dependent increase in IL-10 from macrophages that were treated with BHB. So as you increase the BHB concentration, you increased interleukin 10, the anti-inflammatory cytokine. However, when you look over on the TNF alpha side, there was no effect of BHB on cytokine, pro-inflammatory cytokine expression, suggesting that BHB is promoting an anti-inflammatory macrophage phenotype. So as Dr. Abuelo had said earlier, he talked to you about neutrophil function. They've also done similar studies with BHB, with neutrophils. The main role of a neutrophil is to kill invading pathogens through either phagocytosis, extracellular degranulation, or neutrophil extracellular trap, as Dr. Abuelo had already mentioned. NET, or neutrophil extracellular trap, is when the neutrophil extrudes its DNA to form a trap for the bacteria. I kind of like to think of it as almost like a spider web. When a fly flies into a spider web, it gets trapped and the spider gets to eat the fly. Well, the neutrophil extrudes its DNA to trap bacteria to keep that bacteria from causing an infection. So BHB impaired neutrophil bacterial killing both through phagocytosis, which is on the figure here. It was a dose-dependent decrease, a nice stepwise reduction, all the way up to 8 millimolar concentration, as well as BHB decreased extracellular bacterial killing. What I kind of found a little bit more interesting was the net formation. So this is staining. This is a microscope image where they did an orange staining to look at DNA. So if you're measuring net formation, you're going to look for more DNA to be extruded. And you can kind of see how the DNA looks stringy. And this is the neutrophil expressing net formation. So this panel C is at 0 millimolar concentration of BHB. Panel D is at 1 millimolar concentration of BHB. And panel E is at 8 millimolar concentration of BHB. And as you can see, as you go across these panels, that net formation is being reduced, and that the orange staining that you're seeing here in E is just the DNA that is inside the intact cell, the intact neutrophil. Finally, when you get to panel F, you have a better magnification of what panel E is. And you can see the neutrophils are very much alive and intact. You can see the orange staining here in the live cells. And you can also see the E. coli. So they challenged these cells with E. coli. The E. coli was labeled with a green fluorescence protein. And so you can also tell that the neutrophils are not killing the E. coli. So I like cell culture studies. I think they help us answer research questions without much variation, but cell culture doesn't necessarily, it's risky to extrapolate from cell culture to in vivo studies where there's much more complex biology. So there's been a number of studies that I've done to try to model what we see in transition cows, try to model that negative energy balance. One of these studies was done by Perkins and colleagues in 2002, right here at Michigan State. They took peak lactation primiparous cows and they did an intramammary challenge with LPS. So LPS is lipopolysaccharide. It's a gram-negative, it is part of the gram-negative cell wall. So it mimics a gram-negative pathogen. It mimics E. coli, for instance, without actually causing an infection. It is not a live bacteria. They infused that up into the mammary gland. They had two treatment groups, a positive energy balance treatment group, which is their control, and they also had a negative energy balance group, which is the restricted group, which is in blue, where the cows were restricted for energy intake at 80% of energy requirements for 2 weeks, and then they did the LPS challenge. What was interesting that came out of this study was a delayed febrile response. So this is, they measured temperature over the course of 24 hours. And as you can see, there's a reduction right here at, I believe that is 3 and 4 hours post-challenge in body temperature in the restricted group. And then when you get to 9 and 12 hours post-challenge, there's an increase in body temperature. So there's a delayed febrile response. And if you remember when I was talking earlier about NLRP3, that is a cytokine, or excuse me, that is a mechanism to activate a cytokine that is known to induce fever. So maybe, possibly, we don't know, but that in these cows, there was an elevation of BHB, which caused a delay in that response, which delayed the febrile response. We did an infusion study just recently at Kansas State where we took late lactation cows. There were 12 of them, 6 per treatment group, and we blocked them by parity and by milk production and randomly assigned them to be infused intravenously with either BHB or saline for 72 hours. So here's an image, I hope you can see that, where the cows have 2 catheters in their neck. One is for a pump, which is right here, where we're continuously infusing a solution into these cows. We're either infusing BHB or saline into these cows for a 72-hour period. And at that time point, at the beginning of the infusion, we challenged these cows with strep uberis, another common mastitis pathogen, in both the right rear and right front quarters. The right rear quarter was a quarter that we used for biopsy. The right front quarter we used for bacterial enumeration. We also took blood samples from these cows, but we're currently trapped out of the lab, so we haven't been able to analyze that data right now. So this is a clamp. It's a BHB clamp. Again, here's the pump. Here's a close-up of the pump. Here's the cow with the catheter in her neck with the hose attached to the catheter. We are taking hourly blood samples for 72 hours. Yes, it was painful, and painful for me. We had a team of graduate students and undergraduate students who would help out tremendously, who would take these hourly blood samples to make sure that we clamped the BHB value in the BHB cows. So our target was 1.8 millimolar BHB, and we wanted to make sure that we maintained that concentration as well as we could throughout that 72-hour period. So the black line here is your infusion rate. As you can see, it fluctuates because we're adjusting the infusion to maintain 1.8 millimolar. And then the blue line represents the BHB cows, and the red line represents the control cows. The purpose of this figure is to show that we can mimic ketosis in late lactation cows by infusing BHB into them. These two dots right here, the blue dot and the red square, are the baseline values to demonstrate that there was no difference between the treatment groups prior to the infusion. So then we measured strep uterus colony forming units. To me, this is a really good way of measuring immune function in the mammary gland. So we infused strep uterus into that right front quarter, like I said, and then we took daily milk samples from them to enumerate how much bacteria we could recover in the milk or in the mammary from that mammary gland. And as you can see, during the infusion, there was only a minor change, but as the cow progressed through the lactation, the BHB cows got progressively more severe, and they were unable to maintain that colony forming unit at a low level, unlike the control groups that have a much lower CFU, strep uterus CFU, suggesting that the mastitis was much more severe in the BHB group than in the control group. We also measured vaginal temperature. So we have a data logger up here in the corner that was attached to a blank seeder, so a seeder without progesterone in it. It was attached to the seeder and implanted into the cow's vagina, which recorded body temperature every hour for a 7-day period. I averaged those body temperatures by day. And what you can tell is that the control cows here in red have a nice spike in body temperature, about 39.5 degrees Celsius, or which is about 103 degrees Fahrenheit, on day 2 to 3 post-calving, or excuse me, post-infusion. And then this body temperature resolves over the course of the time of the challenge. The BHB cows, however, are not having that response. Their inflammatory response is, or excuse me, their fever response is almost completely abolished during the infusion. After the infusion is over, they start to mount a febrile response, and we see an increase up to about 39.3 degrees Celsius, or about 102.5 to 103 degrees Fahrenheit, 6 days post-challenge. So as you can tell from this, BHB impairs that fever response. It slows it down, it delays it, which allows then for the strep uterus to grow in greater numbers because we have a delayed immune response. Going along with that, BHB reduced dry matter intake and milk yield during the strep uterus challenge. This was an effective treatment, not necessarily, or excuse me, not an effective treatment by time. So we saw reductions in both dry matter intake during the week post-challenge, as well as reductions in milk yield post-challenge. Kind of surprisingly though, we did not find an effect on milk quality during a strep uterus challenge. So the somatic cell score or somatic cell count was not different between the two treatment groups. Neither was the clinical onset of disease. And by clinical onset, I mean time until we saw clots or inflammation forming in the quarter. However, there was another study done by another group, by Rupert Brookmeyer's group in the University of Bern, who did another BHB infusion study, and they did an LPS challenge. So an LPS mimicking a gram-negative pathogen challenge, an intramammary challenge. And what I want you to get out of this data is that the black triangles here are your LPS BHB cows. The open triangles here are your LPS control cows. And you can see that the black triangles are substantially less in somatic cell count than the open triangle. So the BHB reduced somatic cell count during the onset of this challenge. Now, a lot of times in the dairy industry, we'll talk about how, reducing somatic cell count is a good thing, and it is a good thing, but maybe in the context of the onset of a challenge, at the beginning of a challenge, when we need an immune response to defend the mammary gland, it's probably not a good thing. It's probably quite a bad thing. And it allows for, if this was bacteria, possibly to grow at an easier clip, to grow at a greater rate to cause a more severe infection. Going along with this group also found a greater IL-10 transcript abundance in mammary tissue. Remember when I was talking earlier about mouse macrophages, we saw an increase in IL-10 from those mouse macrophages that were treated with BHB. This data kind of corresponds with that you're seeing an anti-inflammatory response in the mammary gland, which is inhibiting or limiting host immune responses. So just some general conclusions from these studies. BHB impairs innate immune function, has an anti-inflammatory effect, particularly with IL-10. It impairs the onset of fever, which allows for the bacteria to grow, causing a much more severe infection. Particularly this was true with our strep uterus challenge. So how are we going to solve this problem? Well, there are some proven solutions to hyperketonemia. Particularly there's prepartum monensin, or maybe you might know it as rumensin supplementation. Monensin has been consistently shown to reduce BHB concentrations in postpartum cows. And going along with that, it has reduced the incidence of infectious disease. Like I said, I like to describe the transition period as having that domino effect. So it's possible if you reduce metabolic diseases that you can reduce infectious diseases in these herds as well.